Lynch, and I'm an assistant professor of neuroscience in the Department of Psychiatry at Weill Cornell Medicine in New York. I regret that I'm not able to join you at the meeting in person, but I'm grateful for this opportunity to present remotely and tell you about our work using precision functional mapping combined with a dense longitudinal sampling strategy to investigate how functional brain network organization and connectivity differs in individuals with major depression. And of particular relevance for this symposium, I think, will be that the findings I'll share with you today include both trait-like and state-dependent. And I'll start by briefly operationalizing what this term precision functional mapping means. Precision functional mapping is simply the practice of delineating functional brain areas or networks at the level of individual humans, and most often using large quantities of resting state fMRI. This approach stands in contrast, of course, to how functional networks have been mapped historically, which is at the group level, pooling together relatively small quantities of data for many individuals. However, over the past 8 or 10 years or so, precision mapping studies have shown that functional network topology, their size, shape, and spatial locations in cortex, can deviate markedly from what we see in group average maps. In fact, at the individual subject level, there are features of functional brain organization that can be obscured entirely at the group level. And these individual patterns correspond very nicely with task-driven patterns of activity and are increasingly thought of as being trait-like, meaning that they're stable over time in repeatedly sampled individuals and even across different cognitive states. The analogy that I like to use when talking with folks about PFM and why I think it's potentially such a powerful approach, especially in the context of studying psychiatric illnesses that are heterogeneous, is to consider what happens when you take pictures of individual human faces, align them spatially, and then average them. What you end up with is something similar to what you see at the center of the slide, which is an average human face. And you can see clearly what gets emphasized in this type of framework is what is common to individuals and what makes them unique. I think conceptually something similar is happening with our functional network maps when we adopt this kind of group average strategy. All of the rich individual differences that exist across people get blurred or masked out entirely, and instead we're left with something that emphasizes a central tendency of the population but loses a lot of potentially functionally meaningful information. And one of the open questions that motivates the work I'll be talking about today is whether or not there is information in these individual differences that can be used to provide mechanistic insights into understanding psychiatric illnesses like major depression, or help us understand the kind of brain network mechanisms that regulate transitioning in and out of different depressive episodes. To begin to start answering these questions, we started a project a few years ago now, which we referred to as serial imaging of major depression. The idea was relatively simple. We would invite individuals with depression in for weekly fMRI scans, as well as a battery of self-report questionnaires and an interview with a clinical psychiatrist to gauge the severity of their symptoms over the past week. Our goals were to first investigate differences in functional network topology, and specifically we focused on network size relative to what we were observing in a cohort of never depressed healthy controls. And we also wanted to capitalize on the fact that this was a longitudinal data set. These individuals were studied over long periods of time, up to 1.5 years in 1 or 2 cases. And we wanted to ask whether or not there were fluctuations in brain network topology or connectivity that either accompanied or perhaps even predicted fluctuations in the severity of core depressive symptoms. One finding that struck us very early on in this work was the organization of one network in particular being very different in our SIMD cohort. And this network I'm talking about is the Salience network. I'm showing you what it looks like on average in our healthy control sample in A. It's this black system. It's relatively small. In healthy controls, it occupies something like 3% of the cortical surface on average, and it has representation in the anterior cingulate, lateral prefrontal cortex, and anterior insula. In our SIMD sample, we found that this network was nearly twice as large, occupying something like 5% of the cortical surface on average. And here I'm showing you what it looks like in 3 representative individuals from that sample. And in C, I'm showing you the comparison of relative to healthy controls using the full SIMD data set. And on average, the difference was something like 73% larger in the SIMD cohort. We subsequently replicated this effect using 3 independent samples of individuals with depression who weren't studied quite as extensively. They only had a little under an hour's worth of resting state fMRI data, but it was still sufficient to see this expansion in these individuals on average. We also asked whether or not Salience network representation was similarly increased in subcortex and in particular in the striatum, but we found that this was not the case. We also performed a variety of analyses to kind of probe the robustness of this effect. For example, doing split half reliability analyses, so taking individual resting state fMRI data sets for a particular patient, splitting it in half, remapping the Salience network to establish whether or not the topology and connectivity of core Salience network nodes looks similar. And here I'm showing you in 2 representative individuals that was indeed the case. We also explored the extent to which our findings were dependent on our choice of network mapping algorithm. So we used a technique called InfoMap. And what I'm showing you here in 2 representative patients is how similar the maps look when using InfoMap relative to another popular method developed by Thomas Yo's group called multi-session hierarchical Bayesian modeling. We also examined things like the extent to which certain processing decisions like global signal regression impacted the effects we were seeing. And what I'm showing you here at the bottom of the slide is that it mattered very little. Then we went on to ask in what way exactly the Salience network was expanded in these individuals with depression. And to do so, we borrowed this conceptual framework that I'm really fond of, where individual differences in functional brain organization are kind of thought of in one of 2 forms. And the first is an ectopic intrusion. And this is an isolated piece of a network that's located in a very atypical location, kind of like an island of a particular network that's separated from the rest of the system. The other kind of variation is called a border shift, and this is exactly what it sounds like. The boundary between 2 functional systems is shifted atypically either outward or inward. We found that Salience network expansion in depression was overwhelmingly driven by network border shifts that primarily affected functional systems neighboring the Salience network, specifically the default mode, frontostriatal, and cingulopurcular systems. We also found that across different cortical zones, Salience network expansion tended to encroach upon different functional systems. For example, in medial prefrontal cortex, where the default mode network has a great deal of representation, Salience network expansion primarily affected that system. We also saw that across individuals with depression, Salience network expansion tended to affect different subsets of functional networks. Next, we asked if Salience network size was stable over time in highly sampled individuals with and without depression, and we found that it was. Here I'm showing you Salience network size plotted as a function of time for healthy controls and individuals with depression separately. And this is the same finding shown for a single representative individual with depression. And what I want to emphasize here is how similar Salience network topology looks across these individual study visits, even during study visits where this person is no longer meeting criteria for an active depressive episode. This is the same finding shown a different way for 3 of some of the most densely sampled individuals with depression from the SIMD sample. The y-axis is Salience network size, so individual black dots represent different study visits, and the x-axis is the Hamilton Depression Rating Scale score, which is this global measure of depression severity. And in all of these individuals, you'll see there's no relationship between variability in Salience network size and how depressed they were. We also found that there was no change in Salience network size after a rapid-acting neuromodulation intervention called TMS. And in a kind of cross-sectional framework, when considering individual differences in Salience network size across patients and relating that to the overall severity of their symptoms at baseline, we did not see a significant relationship, nor did we see an effect of the number of depressive episodes patients self-reported having during their lifetime. Collectively, the data that I'm showing you on this slide led us to conclude that salience network expansion was not a state-dependent effect. It's not an acute readout of how ill someone's feeling at a particular moment in time. And we instead hypothesized that it could represent instead a kind of neurobiological trait that confers to some extent risk for developing depression. And to test this hypothesis, we turned to the ABCD study and identified children who were scanned multiple times prior to the onset of depression symptoms later in adolescence. And we looked at the size of their salience network compared to other children who did not experience symptoms of depression at any studied time point. We found a similar effect. Their salience network was larger than in children who did not go on to develop symptoms of depression, which we concluded was consistent with this possibility of salience network expansion being a marker of risk for developing a depression. In our final set of analyses, we evaluated if changes in functional connectivity between different nodes of the salience network either accompany or predict fluctuations in symptom severity over long periods of time in our most densely sampled individuals with depression. And that's what I'm showing you here on this slide. At the top, this is a heat map indexing the severity of a variety of different clinical items from a variety of clinical scales that index in one way or another something related to anhedonia. And the x-axis is time, so you can see this person was studied from roughly August 2020 up until December 2021. And the black line at the top of the heat map is a kind of summary measure of how severe symptoms of anhedonia were at any given time point. In B, I'm showing you what their salience network looks like, and in C, this is a summary of how variability in functional connectivity strength between different pairs of frontostriatal salience network nodes relates to fluctuations in symptoms of anhedonia over time in this person. And the connectivity pattern that turned out to be most, that tended to track anhedonia most closely was this connectivity between the nucleus accumbens and the anterior cingulate, which you can see was negatively correlated with the severity of anhedonia. In other words, when anhedonia symptoms were more severe, connectivity between this pair of nodes was weaker. We actually found in this person that connectivity at a given time point actually predicted the severity of anhedonia symptoms at the subsequent study visit in the future. This effect was replicated in part in this second most densely sampled subject from the SIMD cohort. This person was actually receiving ECT unrelated to her participation in our study, so you can see the fluctuations in her anhedonia symptoms are quite rapid in contrast to what you saw in the previous patient, which was kind of a more slow drift in and out of depressive periods of time. Nonetheless, we see a similar pathway being related to severity of anhedonia. Again, nucleus accumbens to anterior cingulate connectivity tracked severity of anhedonia in this person. Interestingly, when looking at other symptoms that tend to co-occur with depression, like anxiety, this accumbens to cingulate connectivity was not significantly related to things like anxiety. Instead, connectivity between other pairs of nodes, and in the case of anxiety, the accumbens to the anterior insula was more closely related to the severity of anxiety symptoms. And finally, I'll note that when using the entire depression sample in a cross-sectional manner and looking at variability in connectivity strength, individual differences in connectivity strength between the nucleus accumbens and the anterior cingulate, we did not see a significant relationship with things like anhedonia, which to us, I think, potentially speaks to the power within subject dense sampling designs for establishing brain behavior relationships at the individual subject. So to summarize, we found salience network expansion was trait-like, meaning it was stable over time, unaffected by fluctuations in mood state, and detectable prior to the first onset of depression symptoms in adolescence. In contrast, functional activity strength between different pairs of cortical and striatal salience network nodes tracked fluctuations in the severity of core depressive symptoms in individuals who were studied over long periods of time in a state-dependent manner. And collectively, we think these findings point to new patterns of brain network organization and connectivity that are neural traits and states, respectively, that are characteristic of depression. And with that, I'd like to thank all of the collaborators and study co-authors on this work, as well as the study participants. And I'm happy to answer any questions or comments over Zoom now. Thank you.